Hello student. Today we start the last beat BSc second year paper 7, that is fish developmental biology. Within that, unit number 1 already we are completed. That is unit number 2, that is developmental biology. Within that, morula, bastula, fat map of bastula, gastrulation already we are completed. The last one, that is hatching and post embryonic development. The last bit of unit 2, paper 7, fish developmental biology. Okay, unit number 2, the last aspect, that is the hatching and post embryonic development of the fish. On my right hand side, there is the figure A, B, C, D. All are the developing stage. That is the early stage during formation of larva. Right. The first point, that is the hatching. While the various organs are being formed, the shape of embryo modify remarkable. The body get elongated and differentiated into a distinct head, trunk, and tail. And sometimes the appendages starts making their appearance, and the yolk sac gets reduced. Finally, hatching occurs. The point is that finally hatching occurs, and the embryo become a free swimming larva. The point is that finally the hatching occurs and the embryo become a free swimming larva. Right. Then on my right hand side, the A, B, C, D, that are the early stage during the formation of larva. Okay. Then the young stage of fish, that is the larval development. The young stage of fish forms the time of hatching. Forms the time of hatching till the transformation into an adult like form is referred to as a larva stage. Hatching means the larva development. How can they form the larva development? The period of larval development varies considerable in different fishes depending upon the fish also development of the larva. A hatching, the larva is usually transparent with some pigment spots of unknown functions. Okay, then notochord and mitome are clear with usually little development of cartilage in the skeleton. Cartilage in the skeleton. A full complete of fins is rarely present, but a primordial fin fold is well developed. In the sectal plane, the mouth and jaw may not yet have appeared, and the gut is a straight tube. The gut is a straight tube. The blood is colorless in the majority of species. Important thing is that in the fishes, the blood is colorless in the larva stage in the majority of the species, and the circulatory and respiratory system are poorly developed. The yolk sac is relatively enormous with presumably hydrodynamic disadvantages. The pigmentation of the eyes is variable, but where the eye is non-functioning at hatching level. When they hatch, that time the eye is non-functioning. It develops soon. As the yolk is resolved, the mouth begins to function. The gut and the eye develop further. Okay, the larva now switch for transfer to source of external food. This is the all the stages already I told. Development of larva stages. This is the yolk sac. The swim bladder. The swim bladder may or may not be present during the larva phase. The fish larva or fry having the yolk sac known as sac fry. Known as sac fry. Okay, it is this one. One figure number one and the figure number two. This is one and two. What is known as that is the sac fry. And after the yolk sac disappear, it is known as advanced fry. What is known as the advanced fry? A fish larva or the fry having yolk sac is known as the sac fry, and after disappear, that is known as advanced fry. Okay. In some species, the advanced fry resembles closely the adult except in size in salmon, trout, and many catfishes. This is the important point. This is referred to as a direct development. Then, on the other hand, in several families of fishes like Lupidae, Cyprinidae, Angulidae, indirect development take place. Direct development take place in salmon, and indirect development take place in Cyprinidae, Angulidae, and Clupidae. In these species, larva undergoes metamorphosis, during which larva characters are lost and the adult feature appear. Okay, adult feature appear. A fry which lose the larva character is like a mini adult and is called fingerlings. Mini mature called the fingerlings. These enter an active feeding stage. Result in the growth of the body, maturation of gonads to become the adult fish. The process is going that the mini mature process becomes the adult fish. The females are usually larger in size than the males, but the latter attain sexual mature early. Okay, post embryonic development. The next bit that is the what we already called as metamorphosis also. Post embryonic development. On my right hand side, that is the metamorphosis. The example that is the tortoise, marshir fish. This is the embryonic development of fishes. Figure number A, B, C, D, and E. Okay. The metamorphosis or the post-larval development, that is the post-larval development may be referred to as metamorphosis. The time to reach metamorphosis may be a matter of days in a tropical species, a few weeks or month in the majority of fish, forms a temperature, latitude, or period of year in the all these things examples of eel fishes, anguli. It is controlled not only genetically but also by temperature and food supply, which may affect the rate growth. Okay, then a clear change that is in the metamorphosis from the larva to the adult forms is reported in many species, while in several others there may even be a less marked metamorphosis, that is salmon and eel fishes. Okay, the swim bladder and lateral line may also develop first at this stage. Then the stage wise, what I'm saying that the stage wise metamorphosis changes in tortoise, that is marshal fish, described below as an example. On my right hand side, that is newly hatched fish larva. Here see eye, brain, pectoral fin developing, spinal cord and the notochord, intestine, and this is the yolk sac. Let it disappear. In the first stage, we can start the stages of morphometric changes, metamorphic changes in turtle. That is stage number one. The larva is yellowish in color with fairly large yolk sac called prolarva. The larva is yellowish in color with fairly large yolk sac and called the prolarva. The yolk sac here, here the yolk sac, all these things. The yolk sac is broad anteriorly and tapering posteriorly with a row of pigments on its upper region, upper and the lower region. That is in the upper region. The larva has broad head pigmented here. The larva has broad head, pigmented eye, and medium continuous fin fold. The dorsal fin is demigrated, but rays are absent in it. All on my right hand side, all are the stages of metamorphosis of tartar fish, that is the ascids. Then stage number 2, chromatophores appear mid-color on the head and yolk sac gets slightly reduced. This is the development. First of all, in the last slide, what can we see there? Yolk sac is full in size, but in stage number 2, chromatophore appear to the head and yolk sac gets slightly reduced. Caudal fin contain 15 to 16 rays. This is the caudal fin. All things have the caudal fins. 15 to 16 rays. The air bladder air bladder starting appearing in the second stage. The pectoral fin remains still in the form of membranous flap. Then stage number 3, the yolk sac is considerable reduced. Yolk sac is considerable the reduced. The dorsal fin is almost complete with rays, but still continue with the caudal fin, which becomes deeply forked, having 18 to 19 rays. Three rudimentary rays appear in anal fin. Pelvic fin is still without rays. Pelvic fin is present but without rays. Then the stage number 4. On my right hand side, all the stages, that is the metamorphosis of tartar fish. Yolk get completely absorbed. The dorsal fin is not confluent with caudal fin. Here the dorsal fin, caudal fin. However, anal fin is having 7 rays. Anal fin here. This is the anal fin, production of anus. However, anal fin having 7 rays still remain confluent with caudal fin. A black spot here mentioned, that is the black spot. Okay, a black spot is seen on the caudal peduncle. Okay, all are the process of metamorphosis of tartar fish, that is the post larva development and hatching. Okay, thank you.